Ladies and gentlemen, welcome to the Australian FameLab National Final of 2025. My name is Dr. Kelsey Poole. I'm going to be your MC this evening. It essentially means I'm going to be politely hassling you back to your seat where you need to be there. To start us off tonight, I would like to welcome to the stage respected Noongar elder and fellow of the WA Museum, Irene Stainton AO, to deliver our welcome to country.
I always have difficulty with that one. Straight in, and then I always have difficulty with this one. It's a bit better. Great. It's lovely to be here tonight, and it's lovely again to be celebrating all of our brilliant young minds and a bright future for our country and in the sciences. I am so jealous because I haven't got a scientific bone in my body and I am so jealous, but I'm glad to be here celebrating with all of you. Kaya, wanju, greetings and welcome, distinguished guests, ladies and gentlemen, colleagues all, and a special welcome to those attending who are live streaming, and as I pay my respects to elders past and present from all of those locations. And a special, very, very special welcome. I can only see one there's a few young people here, but I want to make sure that I acknowledge our young people because they are definitely our future. I'm pleased to welcome you to this location this evening as we're surrounded by significant heritage places. And these places make my people proud when we're welcoming you to the Western Australian Museum, Boola Bardip, which translates to many stories. Firstly, I must acknowledge the creation story of the Waggle and the Derbil Yerrigan, which is now known as the Swan River. Our Waggle dreaming teaches us about the creation of the river, how the bends were made, including where the Waggle laid eggs upriver near Bell's Rapids, then travelled south to shed its scales. It rested in the cool, deep water near the old Swan Brewery and continued on to the Waggle Cave in North Fremantle, where another rest was taken before moving on to the ocean and heading north to Two Rocks. In terms of Two Rocks, there's an associated story about two young people disturbing a meeting of elders at Orchestra Shell Cave in Nirimba. The young people were moved by the elders to another location, and when the tide rose, they became trapped in the ocean and turned to limestone, hence the name Two Rocks. There are many local important places within my run, which include right here where we meet this evening. It was a wetland and a food source. Other heritage places include under the old Bunbury Railway Bridge, which was known as the Hotel of Stars, and along the reed-covered banks of the Derbil Yerrigan, near the East Perth Powerhouse and Claysbrook. These areas were well-known camping places and was used as such up until the mid-1980s, as there was plenty of food and access to fresh water. There was an ochre source not far from here at the Perth Rail Terminal, which was used for ceremonial purposes. And when the Windang Bridge, which crosses the river, was named by local elders as a mark of respect to the rich heritage value of East Perth and to acknowledge traditional leader Yalagonga's wife, Windang. Finally, as I welcome you this evening, I remind us all to think about the importance of this cultural protocol, which has been part of Aboriginal law since traditional times. And once welcome to country, you were invited to fish and to hunt and to share our waterholes. And it makes me sad when I hear criticisms and comments in the newspapers about this being an early thing, a newly created thing. It's actually been there all along, guys, part of our law. But most importantly, being welcomed is about keeping us safe during your visit to Noongar land, and that this message is especially important for visitors from other locations, that in the event of a severe mishap, your spirit will be culturally safe and most welcome in this place prior to entering its dreaming, Wanju Wanju, Nanakaba Nanjaba Buja, welcome all of you to this land. Thank you.
Thank you, Irene. On behalf of the Foundation of the WA Museum, I would like to extend a warm welcome to you and to all our distinguished guests here tonight, including the Honourable Stephen Dawson, MLC, Minister for Regional Development, Ports, Science and Innovation, Medical Research and Kimberley, Sheila McHale, AM, Chair of the WA Museum Board of Trustees, Jason Fair, Acting CEO, WA Museum, Professor James Avanatakis, Director of the Forest Research Foundation, Julie Fallon, Executive Vice President of Woodside Energy, Professor Gretchen Benedicts, Associate Deputy Vice-Chancellor of Curtin University, Professor Melinda Fitzgerald, Deputy Vice-Chancellor of Curtin University, and Professor Ellen de Boer-Woodman, Pro-Vice-Chancellor, the University of Western Australia. Now, it's nearly time to introduce a few more people, and these names you may not have heard before. That's because you're shortly about to meet 12 early career scientists who are only just beginning their careers across a number of different scientific fields. They are the new emerging voices of Australian science who are going above and beyond to communicate their science to not only their peers, but also to industry and general society. Tonight, hosted by the Foundation for the WA Museum in these beautiful surrounds of the WA Museum Boola Bardip's Hackett Hall, they are taking part in the largest public-facing science communication and training program in the world, FameLab. Our contestants have been training for the best part of this year, entering through several competition rounds, including state semi-finals, to reach the Australian finals here tonight. Over the past few days, they have finally met in person for the first time as they undertook a series of extensive masterclasses in science communication. Their goal was to learn to communicate their research in just 3 minutes in a way that every single person in this room will understand. They may be in the early stages of their careers, but I think the average scientist could learn a lot from our FameLab contestants. By undertaking this program and by showing up here tonight, they are demonstrating their commitment to science communication. They are investing in the ability to translate their science that underpins industries across Australia, into something accessible to the average person. It's a skill that I think is going to be critical in this next generation of scientists, and yet one that has been critically undervalued over the past few decades. Science communication is constantly present in our lives. I don't think we always recognise it. This week, for example, how many of you have seen an ad on social media, maybe learned new information through a podcast or radio or TV? If it's involved medicine, conservation, a significant weather event, the environment, food production, very likely it was a form of science communication. We are surrounded by simplified explanations of how the world works, information that is shaped and moulded into a way that we can understand and engage with. Often, without even being aware of it, day-to-day science communication is shaping the way we learn new information, shaping the decisions we make and the beliefs that underpin those decisions. That's a pretty powerful thing. And while being at the forefront of science representation can be really powerful, it can also be really scary. It takes a lot of courage and certainly a lot of vulnerability to make yourself visible, put your image out into the world and open yourself up to critique. I have certainly learned that the image that you present to the world through science communication can and will stick with you. I do a lot of my science in agriculture. Somehow, every time I do something public-facing, I'm wearing this one hat. Headshots, media interviews, this one hat. And the result of this is that without the hat, people they don't recognise me. They haven't seen my forehead, and this here, apparently, is where most of my scientific credibility is. The key, apparently, to my image is the hat. And so I am a science. I'm all about consistency. Is it? Do you recognise me now? Yeah, I can talk about livestock now and you'll believe me. Look, as scientists and science communicators, we do have to ask ourselves these really important questions about their science we're presenting. I just didn't think the question that was really key in my career was, when does this stop being an akuba and start becoming a bit of a personality trait? Tonight, 12 finalists are also putting their image, their science, and themselves out into the world. They have solidified years of work into 3 minutes, communicating with only themselves and maybe a prop or two. Tonight is a reminder that we scientists have the responsibility of not only undertaking research, but ensuring that its impact is translated to communities across the world. With that, I would like to acknowledge that FameLab is only made possible with the generous support of the WA Museum, Cheltenham Festivals, Comet Science Communication Tracing, the Community Broadcasting Foundation, Curtin University, the Western Australian Government through Inspiring WA, the Forest Research Foundation, Murdoch University, Wellspring Foundation, and the University of Western Australia. And of course, our esteemed judges who will be selecting our Australian FameLab winner tonight, who will go on to participate into the international competition. Joining us, we have Deborah Leavitt, Media Executive, WA Museum Trustee and Board Member of the Foundation of the WA Museum, Dr. Jennifer McQuillan, Director, Science at the WA Museum, Lisa Kirkendale, Head of the Aquatic Zoology Department and Curator, Mollusks, at the WA Museum, and Luke Smith, Head of Biodiversity and Science at Woodside Energy. And now, the presentations that we have all been waiting for, our 12 finalists from across Australia. Our first presenter up, we have Anna Antipov with her presentation, Smart Tools and Silver Linings.
Wounds are a pain, literally. And this, a smart gel we've created using cutting-edge nanoparticle technology, is the new tool we're going to use to solve this problem. Let me tell you how we got here. I want you to think of wounds like an open zipper. Usually, when you get a small cut or burn, your body works to close the zipper and the wound heals. But in the case of chronic wounds that are slow to heal, the zipper gets stuck and stays open, sometimes for over 12 weeks. And whilst the zipper's open, it means nasty bacteria can get in and cause infection. You might be thinking, so what? Just use antibiotics. They've done the job for over 100 years. Well, as the saying goes, if the only tool you have is a hammer, then everything looks like a nail. The problem with antibiotics is they've been used so much that bacteria have evolved. And this tactic no longer works. They've caught on. Since the hammer has failed, we now need a screwdriver, something new that does what antibiotics used to do to stop once treatable infections becoming deadly threats. Currently, our best alternative is to use silver. Yes, the same silver metal that makes your jewelry, but to fight infection, we create teeny tiny particles of silver that can be added into dressings or creams. This has been effective so far, but now we have the problem of having too much of a good thing. When silver is applied on large areas bigger than the palm of your hand, or for longer than a couple of weeks, it can build up in your body, become toxic, and cause some serious damage. So we need a way to deliver silver in a clever, controlled, and dynamic way. How do we do that? Using smart technology, of course. Did you know that when an infection occurs, there are some important changes that happen? Wound temperature actually increases, and the wound environment becomes less acidic. Now, Remember our smart gel? The reason we call it smart is that it relies on technology that is able to respond to these signals from our body and release the silver particles only when they are required, preventing the risk of toxicity. What's really cool is that we've shown that our treatment is just as effective at fighting infection as other treatments that are already available, but using 15 times less silver. And not only that, it also helps to promote wound healing, helping to close the metaphorical zipper fully and quickly. So as we continue to take our new tool out of the lab and into the real world, the next time that you get a wound, remember that although it is a pain, healing it doesn't have to be.
Thank you, Anna. I wish my science was that sparkly. Judges, on to you for questions.
Oh, is that working? Okay, excellent. Great talk. I suppose I love how you're looking at an issue. Maybe you can just talk a little bit. Why do we need to find a difference to antibiotics, and can we actually use this to go both? It sounds like you're doing the outside in with the lining on the outside. Is there still a role for antibiotics to fight from both the inside and the outside?
Great question. So yes, our treatment is topically applied onto the surface of the wound, and it's really great because our gel will set inside the wound and can really get into the deep crevices. Right. The issue with antibiotics and antimicrobials is that there is such a quickly developing resistance, and simply new antibiotics are not being discovered fast enough. So at some point, if we don't use alternative solutions, they will be unusable, even in hospital settings. So we really need to focus on finding alternative treatments so that doesn't happen and that we can keep everyone safe and happy and healthy.
Great, thanks.
So Anna, can you tell us what your sort of the aha moment was sort of during your kind of research journey?
Could you rephrase?
Well, have you had an aha moment, a sort of a moment where something's kind of coalesced, a point in your research where, yes, there was a light bulb moment, I guess, which has helped you sort of go, yeah, I'm onto something here?
I feel there's been quite a few of those. I'm very fortunate to work with a great team that's been very involved in the development of this product. We've had a lot of collaborations from chemists, material scientists, industry partners, right through the testing phases. So from the initial conception of this is something that we can make that can really solve and address an issue, right through to the different stages of testing of yes, this is working, yes, we're doing well, refining the formulation, to now being at human trial stages, which we're trying to get funding for so that we can really actually get it out into the real world and hopefully see it making an impact.
Our next finalist is Kenya Fernandes with her presentation, Bees Just Want to Have Fungi.
This is Beatrice. She's a native Australian bee. She's about 6 weeks old, which in bee years makes her a senior citizen, and she's about to have a rough day. This morning, like every morning, she's getting ready for work. She shakes out her stripes and hits the skies. But something's not right. Her favourite patch of wildflowers, it's gone, replaced with a parking lot. And the weather, it's hot, cold, wet, all on the same Tuesday morning. Exhausted, she stops for a drink of water in someone's garden. It tastes chemically. Beatrice doesn't know it, but the owner sprayed pesticides yesterday. She suddenly feels sluggish, disoriented. This is where Beatrice's story usually comes to an end. Another casualty in a silent crisis that's playing out across Australia. Our pollinators are in serious trouble, And when they suffer, so does everything they support: our food, our flowers, our ecosystems. It can feel like a losing battle. But what if instead of fighting harder, we needed to look closer? What if Beatrice already had allies that we just never thought to look for? Allies that we're usually scraping off our bread or scrubbing out of our showers. I'm talking about fungi. Beatrice is surrounded by these microscopic passengers, fungi that live in her body, in her nest, and even in the food she eats. And we're just beginning to understand all of the ways that they might be helping her. Some fungi make natural medicines that help Beatrice fight disease. Others boost her immune system or help her process her food. My research is about understanding how these partnerships work. I'm mapping which fungi nourish bees, which ones protect them, and how bees use their own natural pharmacy of honey and plant resins to support the growth of the good fungi while keeping the harmful ones in check. And this isn't just a one-way relationship. Every time Beatrice visits a flower, she's not just collecting nectar and pollen, she might also be picking up and spreading beneficial fungi, moving it from plant to plant and back to her nest. So it's not just bees helping plants, like we once thought. It's bees helping fungi, helping bees, helping plants, helping ecosystems. It's a symbiotic spiral of success. We're on the verge of understanding these relationships in ways that could change how we protect our pollinators. Instead of just fighting against those threats, we might be able to strengthen the ancient natural partnerships that have helped bees thrive for millions of years. So the next time you bite into a juicy piece of fruit or spot a beautiful native flower, take a moment and thank a bee like Beatrice. And maybe thank a fungus too.
Beautiful presentation.
Stop.
Judges, your questions.
Hello, that was great, thank you. Can you tell us what is the one thing you want us to take away from your presentation today?
So I would love for everybody to know that there are so many more bees out there than just the European honeybee. Australia is home to just an incredible biodiversity of native bees. We actually have 2,000 native species, and many of them are amazing pollinators of our native Australian plants. So if you want to do something to support our bees, you can learn more about the local ones in your area. You can plant some flowers in your garden, go pesticide-free, and a lot of our native bees are actually solitary, so they make their homes in patches of dirt or old pieces of wood. So if you've got something like that in your garden, resist the urge to over-tidy and leave some of those wild spaces for our bees.
Awesome. Thank you so much. That was a really powerful presentation. I just wondered a little bit about the power of the prop and how you developed kind of the props that you used in your presentation.
Yeah, so I think the power of a bee and making it a character that you can relate to is so important, because I think, there's so many environmental messages that we're hit with all the time, and as humans it can be very depressing to try and care about all these things at once, but to try and put a face to it and make it something that you empathise with, I think, is something really powerful and strong. Face to the fungi. Thank you.
Our next finalist is Riley Easton with his presentation, MicroRNA, Small Molecule, Big Impact.
In 2003, scientists mapped the human genome, the instruction manual for all humankind. And it's jam-packed, full of all the information you need to know to make your very own human. The information on the pages are your genes. And just like pages form chapters in a book, your genes work together in small groups to form an amazing final story. The story of you. Hang on a second. Why is this page blank? Oh no. This whole chapter's blank. I wonder what it was about. The title says, How Your Body Regulates Literally Everything It Does and Makes Sure We Don't Die. That's not good. Someone should probably research this. Well, it's a good thing I'm here. Now, as that title subtly hinted, the genes that should be on these pages are really important, and they're called microRNA. But hang on, if they're so important, why aren't they in the book? Well, microRNA are a bit different to run-of-the-mill genes. When you think of your genes, you probably think of different characteristics you have, internal or external. But microRNA don't make any of these characteristics. Instead, they regulate how your other genes work, turning them on or off. Your microRNA are your body's microscopic micromanagers, and they're really good at their job. And so, to help people learn more about them, our scientists have been working on a sequel. And it's full of amazing stories all about your microRNA. Let's have a read of one of mine. It's called You, the Flu, and 652. It starts off with you, our damsel in distress. And you're being chased by an evil monster. Oh no, it's the flu. But never fear, your hero is here. It's microRNA number 652. 652 and the flu come together in an amazing final battle, and then, I don't know. I haven't quite figured out that part of my research. What I do know is that without 652, we become vulnerable to the flu. We just don't know how or why. 652 is a microRNA, so it must be regulating a pretty important group of genes. If we could figure that out, it would mean big things for how we design antiviral therapies. And this is just one of your body's microRNA. Humans have over 2,000. I wonder what else microRNA could do. Guess I better get back to work, because this story is only just beginning.
It's the little things indeed, Riley. All right, judges, please, your questions.
Great presentation, loved it. So you've done book 2 now. You're cast yourself 40 years ahead. You've done book 2 and you've discovered that you can use RNA to help with the flu. What does that look like for someone in that future? How are they getting administered that, and what does it look like through that treatment?
Yeah, absolutely, great question. And RNA is, we all remember COVID, and the mRNA vaccine was huge, a huge leap forward, Nobel Prize winning leap forward in vaccine technology. And so what microRNA kind of looks at doing is going another rung higher on the ladder, earlier, and a better immune defense in people. mRNA is above the protein vaccine, microRNA, we're going one rung higher.
So, Riley, can you explain to us what's the biggest benefit that you've got so far out of your FameLab journey?
It's hard to pinpoint one. I think one of the best things you'll see 9 more amazing science communicators, and you've seen 2 already before me. Just getting to meet all these amazing other scientists that in my little microRNA niche at my university, I probably never would have gotten the chance to meet. And now I'm learning all these incredible things like bees, and I won't spoil the rest. But they're an incredible group of people, and I'm so glad to call them my colleagues and friends.
Our next finalist is Minduli Withana with her presentation, Treating Multiple Sclerosis One Spark at a Time.
Picture this. You wake up tomorrow to a world gone silent. Every single connection in your home has been destroyed. There's no flickering of lights, no glow from a screen, no way to call for help. You're trapped, and the invisible lifelines that connect you to the outside world have been slashed brutally. This is not a dystopian nightmare. It's the harsh reality within the bodies of people suffering with multiple sclerosis. MS affects over 2.8 million people worldwide, mainly young people that now have difficulty walking, talking, seeing, even thinking. But what exactly causes this? You see, just like this cable conducts electricity, your nerves carry precious signals that keep you living. Surrounding each nerve is this protective coating or insulation that we call myelin. In MS, however, your immune system strips this insulation away. Without it, your nerve signals stop moving forward. For decades, we managed this issue by suppressing the immune system. Unfortunately, this leaves people vulnerable to infections, kidney failure, and even cancer. So I asked myself a different question. How can I help the body repair its own broken wires? This led me to a tiny but powerful protein, something that we think could act as the body's own electrician, something called the PAC1 protein. In the lab, I studied what happens to this insulation in the presence or absence of the PAC1 protein. So, what did I discover? When PAC1 is not there, this insulation breaks down much faster. So then I asked myself, what if I just picked up the phone and called the electrician back up? What if I switched PAC1 back on? And this is when I was truly surprised. It was like watching a broken cable suddenly rewire itself. Balance and coordination improved dramatically. Cells in the brain looked far less stressed out. So the next time you get home and the lights flick on without drama, take a moment to think of those whose bodies aren't so coordinative. Because perhaps with the right tools and just a little bit of science, we may be able to help them find their signal again.
That was a
Great presentation. Judges, over to you for questions.
I'm jumping in. That was fabulous. I'm really excited for your research. Just wondering about forecasting 10 years ahead. What does it look like? What's the next step? Clinical trials, where do you go next?
That's a great question. So that's definitely the end goal in my research, but at the moment we're of course quite early on, so we would need to go a long way down the pipeline before we could take this to human trials. Right now I'm really interested in how PAC1 actually does this job, how it protects, but at the same time, how can we sort of help it grab that toolkit and get to work repairing that insulation? But yes, definitely something we're hoping to do down the line, and we have hope for that.
Thank you. Tell us a little bit about why the immune system is so interesting to study, especially from your own point of view. What was it that sparked your interest in this particular topic?
Thank you for that question. What is interesting to me is the fact that as humans, we're living beings walking around functioning in the world. We expect something like the immune system that's evolved to protect us, to keep us safe in life, to, have an efficient function. However, in MS, you find that it's the immune system that actually causes this disruption, causes this damage. And so to sort of see a way to work around that, not just try and sort of dampen the immune system, but to help stop the progression of disease by identifying new things that we can target is really exciting to me. And that's sort of what drew me to this research, because I thought, okay, if the immune system's suffering, how can we help target this in other ways and help treat people? Thank you so much.
Our next finalist is David Stewart-Yates with his presentation, Flies, Forensics and Fingerprints.
How many of you have ever wanted to be just like a fly on the wall? Listening to conversations, spying on people, or even witnessing a murder? Well, forget the police. Flies are often the real first responders, arriving at the body within minutes. Only for them, it's not a crime scene at all. It's dinner and a perfect place to raise their kids. They lay their eggs, which become maggots, then pupae, and finally the adult fly. A predictable cycle, like how a caterpillar will cocoon and turn into a butterfly. But this cycle isn't just a cycle. It's a biological clock that can help us uncover the truth of this crime scene. When the fly arrived can tell us when the death occurred. The type of fly present can tell us if the body's been moved, and the fly's stomach contents may even reveal that the victim's been poisoned. But before we can get any of these answers, we must know two very important things. One, the species, and two, its age. Get either of these wrong, and we risk framing the wrong person. To identify the species, we use a microscope. The problem is, what if the sample is damaged? Key features are now missing. My answer for a more reliable approach lies in the fly's fingerprints. And no, I don't mean the fly's 6 legs. A fly is coated in a unique blend of chemicals, forming a chemical fingerprint. For them, this is like a chemical ID badge. They use it to identify the species, the age, or even the gender of another fly. But I can use it to reconstruct the events of the crime scene. Here's how it works. I'll collect the fly from the scene and place it into a vial. I then add a special liquid to extract this chemical fingerprint from the skin, which is then read by a machine. From this, I can identify the species and its age, allowing me to determine when this fly arrived and estimate the time since death. But this one chemical fingerprint also reveals so much more information. Its chemical composition will vary with geography and exposure to poisons, allowing me to track body movement from this scene to another scene, or even get insights into the cause of the death. And because we only need a very small piece of this insect, it can work on fresh samples, as well as century-old and damaged samples, opening up opportunities to investigate 100-year-old cold cases, war crimes, and even historical mysteries, revolutionizing criminal and legal investigations around the globe, all from the chemical fingerprint of the fly.
Thank you, David. I will not be looking at flies in the same way again. Judges, your questions, please.
Obviously, we have a huge fascination with death and solving who done it. I'm just interested, how did you get involved in sort of the CSI science that we all are fascinated by? What was your journey to get there?
My journey involved me actually being told by my mother after telling her kind of what I was interested in school, and she was like, that sounds like forensic science. So I was interested in doing forensics before I even knew what it was, but I've always loved biology, chemistry, and then the extra problem solving that comes with a crime scene with no case is the same. Absolutely loved it.
So David, great presentation, thank you very much. So in your work, how has forensic science evolved beyond this sort of stereotypical kind of notion that we have around, which is, often popular in the modern media?
Sorry, can you please repeat the question?
So how has forensic science evolved? A lot of us think about forensic science. We think of CSI, that kind of thing. A lot of us have watched it on TV. How has it evolved? Do you think?
Evolved. Sorry, yes. I thought you said involved. No, sorry. It has absolutely evolved. So as we get new funding and we get better techniques and understanding of the factors that may influence our evidence, we can then adapt and evolve. And especially for this case, using the chemical fingerprint, it's a lot more stable than the DNA, so it allows us to increase our window of detecting and trying to interpret this case.
Our next finalist is Nimllash Thangam with The Invisible War, One Fungus, Big Consequences.
Hello, everybody. What if I told you A bowl of soup you're having today is only possible because of a tiny battlefield happening in every farmer's field between a plant and a fungi. Lentils are pulses, like peas. They are packed with protein, nutrients, and other essential amino acids, which are essential for our human body. For millions of farmers and people, it's just not food. They are just a way of life. Now, just imagine this. When this not so tiny lentil plant anymore, minding its own business, happily growing in the field, bam, along comes this nasty fungus called Ascochyta. It is kind of like a Voldemort of lentil diseases. It crosses an ugly brown spot, weakens the plant, and when things gets bad, it wipes out the entire crop. This is tragic as it sounds. But don't worry. This is where I come in as a plant pathologist, working closely with the plant breeders. We are like a matchmaker for plants. Our job is to find the lentil that are strong, that can withstand this fungus, and then we breed them with other tasty, productive lentil. But it's kind of like a Tinder, but for plants. Swipe right for resistance. But this process takes time, even decades. We test hundreds, sometimes thousands of varieties to identify that one plant that can withstand this fungus like a tiny gladiator. And then we grow them in the field, we grow them indoors, and we even throw a fungus at them to see who survives. And we also speed up this process using molecular tools like DNA markers. It is kind of like a Google Maps for genes. Its job is to identify the lentil plant which are resistant before even the symptom appears. But this resistance alone is not enough, because similar to COVID and the flu, this pathogen evolves as well. It overcomes the resistance, and it even creates a resistance for the fungicides. So constant monitoring of the population of this pathogen in the field to find the mutation in the pathogen, and then also along with the disease resistance, we'd be able to manage this fungus, and we stay one step ahead of this pathogen. So the next time you're enjoying a nice bowl of lentil soup, just think it is just not food. It is the part of science, dedication, and lots of fungus fighting love. So I would like to finish by saying we are not just fighting a fungus, we are feeding the future.
Thank you, Nimllash. Love that energy. Judges, your questions.
I think that's the line of the night, Tinder for plants. Can you tell us a little bit about what you think the impact for the industry would be with your research?
The impact for the industry is billions of dollars, because currently Australia is one of the leading producer of lentil, and it's exporting #1 lentil exporter in the world. And when this disease strikes, it can almost cause about 50% of the yield loss when the things are very, very bad. So it can cause billions and billions of dollars for the farmers. That's the impact it can cause.
Thank you.
Thank you so much. Wonderful talk. How do pathogens in pulse crops like lentils affect both agricultural sustainability and human nutrition globally?
I think both are interconnected, I would say, because as the global population is increasing currently, we are looking for more sustainable farming where we can sustain both the population as well as we can farm a climate-smart agriculture. So when this disease as a biological entity come into play, it causes a broken system in the supply chain, which causes an outrun similar to the COVID you have seen. It has changed the whole market of things in the field. So this disease can cause a very impactful thing in the future if it's not managed correctly. Thank you.
Our next finalist is Tammy Lee with Atypical Embryo.
How many cells do you think you're made of? It's tens of trillions. But once, you were just one. Everyone's story starts the same way, with the egg and the sperm. They lock eyes, sparks fly, and fertilization. These are definitely not to scale, by the way. The egg is 10,000 times larger than the sperm cell. I work with these tiny human cells in the lab of a fertility clinic because for some hopeful parents, science plays a role in growing their families. The egg and the sperm form an embryo. And in the lab, I get to watch embryos grow through time-lapse videos. It's like watching the world's slowest movie ever called embryo development. Let's zoom in. We expect to see a cell of an embryo divide from 1, boom, into 2 cells, then 4, 8, and so on. But this doesn't always happen. I've seen a cell of an embryo divide, but then merges to 1 again. Wait, what? That's not in the textbook. I've even seen a cell of an embryo divide, but into 3 cells, not 2. Definitely not in the textbook either. As these funky embryos don't follow the textbook, they're often overlooked and even discarded, as they're assumed to have the wrong number of chromosomes. But for me, I wanted to know, do these funky embryos have potential? And are we dismissing those that deserve a chance? Because for that hopeful parent, the funky embryo might be all they have in the lab. Their one chance at having their baby. So, for my PhD, I watched the movies of these funky embryos, frame by frame, and some of them can make it. They can self-correct by removing abnormal cells from forming part of the embryo entirely. They can implant and become healthy, beautiful babies. Embryos are incredible, but they don't always follow the textbook. And maybe that's the most human thing of all. Things may not always go to plan, but we can still become something extraordinary.
Thank you, Tammy. Best use of Play-Doh that I've seen to date. Judges, ask away.
Thank you, Tammy. That was fabulous. So you mentioned that you work in a fertility clinic. So my question is really around how your research helps families who face the dilemma of infertility.
Absolutely. So my research has the potential and is having the potential, we are finding from our results, we can improve embryo selection. So traditionally, embryos that have atypical appearances and behaviours, as I said, they might be overlooked, but we are finding from my research that they can indeed lead to healthy live births. And also the ones that are able to self-correct, they lead to higher live births as well, so it will improve embryo selection in the lab for us. And as a result, it shortens time to pregnancy for our patients.
Thanks, Tammy. Great talk. I'm interested about your journey to think or want to do a PhD in that first few cells of our existence and actually helping them. What drove you to looking at this part of science?
Yes, so great question. So if we're getting a little bit personal, I myself was created in the lab, so I am a test tube baby. So really, I like to call it the circle of life. It's the reason why I'm standing here. It's why 1 in 16 babies in Australia are born. It's why, it's incredible. So I guess that's how I got on the path that I'm at, and I just really want to improve the outcomes for our patients.
Our next finalist is Caitlin Dudley with her presentation, Goldilocks and the Three Beans.
Once upon a time, there was a farmer called Goldilocks, but she wasn't looking for porridge. She needed a sustainable crop that provided protein, fibre, and carbohydrates. So she wanted some mung bean. But not just any mung bean. She needed mung bean that flowered just right. However, in this story, I'm actually Goldilocks. This isn't a fairy tale. This is my research, and I study what makes mung bean flower on time. And in this story, these are my three beans. My first mung bean flowered way too fast, right in the middle of summer when it was too hot. All the flowers wilted off, and I barely got any pods. My second mung bean, this one was too slow. It missed the warmth of summer, and it barely bloomed at all. But my third mung bean here, this one flowered just right. Now, in agriculture, timing is everything. If plants flower too early or too late, they can't make the most of their environment. And for farmers, this means lost yield, lost income, and so many lost opportunities. Now, what's the secret behind this timing? Well, one of the most important ones is photoperiod sensitivity, and that's how plants respond to day length, because plants need to time their lives in accordance with the seasons. Now, for mung bean, some of them really like short days. Other types, they don't really care. But for most mung bean we grow here in Australia, we don't know what they like. So I've been looking for answers. Goldilocks didn't settle for that first bowl of porridge. She tried, tested, compared, and figured out what worked. And that's exactly what I did, using genetics light and mung bean, of course. I grew almost 2,000 different mung bean lines in several field trials in southeast Queensland. They were descended from 30 diverse parents that had been collected from key growing regions all around the world. Now, in the field, I saw a huge range in flowering time, and I found out that this was driven by both genetics and the environment. So to make sure that photoperiod was actually involved, I grew 12 of the most diverse parents in glass houses where I made the day length 8, 12, and 16 hours long. Now, for some of my genotypes, like these two, they didn't flower at all under long days. And surprisingly, they struggled under short days too. But for other genotypes, like this one, it flowered reliably, the exact same time, every single treatment. And the cool part is, I was able to discover parts of the mung bean genome, that DNA instruction manual, that control flowering time. They're called QTL, or quantitative trait loci, and you can think of them as the genetic switches that turn on flowering. Now, these three mung bean, they're not just props, they're real possibilities. And by understanding the genetic differences, we can create new varieties that flower reliably, no matter the season, no matter the location. So next time you hear the name Goldilocks, don't think porridge, think mung bean.
Great presentation. Judges, over to you for questions.
Thank you so much. Your shirt is fabulous. I believe that's a prop. So we've heard about lentils, and just wondering a little bit about how important mung beans are to Australia and the global economy and agricultural.
Yeah, so mung bean is a crop that's quite popular in Southeast Asia, and it is really important there for providing protein and fibre. We call mung bean an orphan crop, which means it's regionally very significant, but it's kind of forgotten on the research and market scale. So it's really important for different communities that we actually put more research there. And for Australia, it's become a really important summer cash crop. So farmers like to grow it, and especially as a legume, it puts nitrogen into the soil to improve the quality, and then we export it.
That was so interesting. What is it that would help you the most in advancing the research that you've done now?
Well, I think just a lot more invested resources in mung bean and orphan crops in general, because they're often kind of the little forgotten cousin of research sometimes. So I think trying to remember them in the other research that we do, and sort of a lot of collaboration could really work.
Our next finalist is Aayushi Khillan with her presentation, One Loop at a Time.
You know, kids these days are into their iPads and TikTok, but when I was little, I had this. The humble slinky. It was peak entertainment. I could watch it go down the stairs for hours and hours. And when it works well, it has this beautiful rhythm. Stretch, release, stretch, release. It actually reminds me of how our brain talks to the rest of our body, sending signals smoothly down nerves all the way to our muscles. That's what allows us to walk, to wave, to wiggle our toes, whatever we want to do. But what happens when that signal gets tangled and twisted? Well, now the messages our brain tries to send becomes delayed and super jerky. This is what happens in a condition called cerebral palsy. Now, cerebral palsy occurs when there's damage to a little baby's brain, often around childbirth. And this damage scrambles the way that the brain and the body communicate. So just like that tangled slinky, these kids end up with body movements that are stiff, jerky, and super unpredictable. But here's one thing a slinky can't show you. About 3 in 4 of these kids experience pain every single day. It's not hard to imagine that if your muscles were constantly sore and stiff, you'd be in pain all the time too. And for these kids, it's their daily reality. That pain steals sleep, anxieties, friendships, school days. It fuels frustration, isolation, and worst of all, it often goes unnoticed. And even if we do notice it, we don't know what kind of pain they're experiencing. See, not all pain is the same. In fact, research suggests that there's 3 types that could be affecting them. The first is from those tight, overused muscles. The second could be from damage to their pain signaling system. And third, their brain could just be sending false panic alarms when actually nothing painful is going on at all. Right now, we treat these kids completely blindly. We throw random medications, surgeries, therapies, just hoping something randomly helps them. It's like yanking at that tangled slinky, hoping it magically unravels. Well, spoiler, it won't. That's where our research comes in. We're using sensory testing and clinical tools to decode that pain layer by layer. We're asking questions like, well, what kind of pain is it? Where is it coming from? And what treatments might actually help them? If you remember just one thing from my speech, let it be this. Access to adequate pain treatment is a fundamental human right. But how can we treat your pain if we don't understand what it is? Kids with cerebral palsy aren't broken. They're brave, they're strong, they're super resilient. Yes, their slinky will forever remain tangled, but with patience, science, and the right approach, we can stop the pain that's holding them back, helping them spring forward one loop at a time.
Thank you, Aayushi.
Thanks.
I miss the slinky days too. Judges, your questions, please.
Thank you very much, Aayushi. That's fabulous. I have a friend who suffers from cerebral palsy, so it's great to hear that you're doing this kind of research. So what are some of the biggest challenges in accurately measuring pain in children that you face, or you and your other members of your research team face?
It's so difficult. Pain as a concept is difficult, first of all. What is pain? Your pain versus my pain is completely different. You know, we often use this pain scale in the clinic. Oh, tell me your pain on a scale of 1 to 10. What is a 10? What is a 1? It just doesn't make any sense, let alone to little kids. On top of that, in kids with cerebral palsy, a lot of them have cognitive disabilities. A lot of them on the other side of the spectrum can't even speak. So then it gets to, well, if they're not speaking, how do we know they're in pain? So there's a lot of difficulties, but we're trying to at least take one step at a time to get somewhere, because right now we're not even acknowledging that they are in pain, let alone doing something about it.
And then I suppose the lead-on question, great talk, is, so the idea is to understand what sort of pain, and then what is you have different treatments. Is that what it looks like in the future? Is you have basically a cookbook of different treatments depending on the pain?
Yeah, that's a great way to put it. In a world of sort of precision medicine, our goal is to target a treatment based on what they're experiencing, right? Rather than just throwing something out in the dark. So there is different types of pain, and our goal is to understand, well, what kind are they experiencing, and can we maybe tailor our treatments to be more specific to that kind of pain? So at the end of the day, we hope that they get a better outcome from it.
Our next finalist is Drew Cylinder with his presentation, Worms on Drugs.
Let's say you're in hospital. You've just woken up from general anesthesia. Your surgery went great. But that night, you see something strange. The walls are moving. The floor is squirming. You look down at yourself. There are worms crawling up your chest towards your mouth. You ring the nurse, but they come in and they say, there's nothing wrong. That's because there is nothing wrong. There are no worms. You've hallucinated the entire thing. Now, this can actually happen. Hallucinations are a symptom of post-operative delirium, a potential side effect of general anesthesia. Now, when general anesthesia works, which it almost always does, it can feel a little bit like going to sleep. But sleep and general anesthesia are very different things. I'll give you an example. If you were asleep and I stabbed you with a knife, you'd wake up, right? This wouldn't be the case if you were under general anesthesia. Under general anesthesia, you can't move, you can't feel pain, and you can't form new memories. Now, these are the effects that we want, but general anesthetics can have many side effects. For example, problems with the heart and lungs, as well as that post-operative delirium we just discussed. So wouldn't it be great if we could find some drugs that could reverse the effects of general anesthetics, both good and bad? Here's the catch, though. Even though we use general anesthetics all the time, we don't really know how they work. And they remain one of medicine's biggest mysteries. So, in order to understand this further, I've gone down to the most basic unit of our nervous system, our brain cells, also called neurons. Now, I want everyone to take two fists like this. I want you to put them together. This is about how big your brain is. And packed into that two-fist-sized space are about 86 billion neurons. That's a lot. That's way too many for me to study. So instead, I use a much simpler organism. This is a nematode worm. This one's not a hallucination, don't worry. In real life, though, these guys are only 1 millimeter long. But in that tiny body, they have neurons just like you and I. These have been genetically engineered so their neurons are different colors. This allows me to see where the neurons are in the body. They've also been genetically engineered to light up when they're active. This allows me to see how patterns of neuronal activity change under general anesthesia. What I found is that general anesthetics don't necessarily turn neurons off. Instead, they cause neurons to miscommunicate. And we are now trying to find drugs that can reverse this effect of general anesthesia. So in my day job, I give a lot of worms a whole lot of drugs. But I'm doing this because most of us will undergo general anesthesia at some point in our lives. And I think that when it comes to solving one of medicine's great mysteries, this tiny worm has a big part to play.
Thank you, Drew. Great presentation. I'd love to know what's going to happen to this worm after your presentation. Where's that going to live?
I'm not entirely sure. I might donate to FameLab or something. We've all named him Benito. His name is Benito.
Worthy cause. Over to you, judges, for questions.
Hi, Drew. That was really fun.
Thank you very much.
I enjoyed hearing that. I wonder if you've ever been under general anesthetic.
I have, and luckily for me, it was a pretty regular experience. There were no kind of side effects.
So it wasn't the spark for your research?
No, it wasn't the spark for my research, no. The spark for my research really comes from the fact that general anesthesia is something that applies to all animals. It doesn't matter how big the animal is. You could be a cassowary, you could be an elephant, you could be a fruit fly, you could be a worm. It doesn't matter. General anesthetics will knock you out at about the same concentration, which tells us that general anesthetics are affecting something fundamental to how our brains function. And if we can figure that out, we can learn a whole lot about the brain.
The prop is fabulous.
Thank you very much. I spent all Sunday sewing this. It's been an adventure.
That was one of my questions, is how hose have gone into that, hosiery have gone into that. What's the next step in your research? What is...
So I mentioned in my talk, we're looking at these drugs that can reverse general anesthesia. And right now, we're looking at a whole bunch of these different drugs. And what we found is really interesting is that, let's all imagine that drugs have a shape, right? We found that the drugs that are most effective at reversing general anesthesia kind of have this similar shape to general anesthetics themselves. And we think that's because these reversal drugs are competing with the general anesthetic in your body. Thank you very much.
Our next presenter is Faris Ruzain with dating advice, bring shiny rocks.
They say a painting is worth 1,000 words. If I showed you this painting that I made at age 5, the words that come to my mind are, my baby boy's an artist, as per my mom. Now, the only reason I could remember any of that was because my mom recorded everything, including the date behind this painting, which helped me remember the context behind it all. Now, what if I showed you these ancient paintings found on Balangara country in the Kimberley of Western Australia? What was the context that went behind them? Well, much like my brilliant art at age 5, knowing the ages to these paintings can tell us a lot about what was going on when they were first made. So, how do we do that? Well, archaeological scientists like me use a technique known as radiocarbon dating on shiny rocks like this one that are found either above or below the paintings to tell us how old they are, because you see, we can't use radiocarbon dating directly on the paintings because they're usually made with pigments that contain little to no carbon at all. But wait, hold on a minute. What is radiocarbon dating? Let me explain. Every living thing, including you, me, and this tiny bacteria on my fingernail, are all absorbing carbon from the environment right now. However, the moment that organism dies, the clock starts, and carbon will start to decay. You see, carbon decays by half roughly every 6,000 years, and scientists like me pick up on whatever carbon that's left to trace the time back to when that organism first died to know how old something is. So, am I then saying that these shiny rocks were alive? No, not exactly. But my research has shown that these shiny rocks were actually made by bacteria thousands of years ago, and they have left traces of their carbon within the rocks that we can then use for radiocarbon dating. This means that the dates that we get out from the shiny rocks are reliable, and by extension, the dates we put onto the paintings are reliable as well. So now that we know how to date ancient rock art, so what? Well, like I said at the start, knowing the ages of these paintings can tell us a lot about what was going on when they were first made. For instance, by tracking climate records, we could tell that people were painting more pictures of animals like this back when sea levels were low and people had more access to the lands and animals. But over time, we noticed a shift in that people started to paint more pictures of humans in ceremonial dress. And this coincided with the time of when sea levels were high and First Nations peoples had to move inland to form closer-knit communities. Now, they say a painting is worth 1,000 words. And my hope is that through radiocarbon dating, we can add more words not just to these incredible paintings that have stood the test of time, but also more words to the lives of the real artists, the First Nations peoples who painted them in the first place.
Brilliant job, Faris. Judges, over to you for questions.
Thanks, Faris. So am I correct in saying that the technique that you've developed using bacteria means that you don't have to destructively sample in order to get correct dates?
That's a very good question. Unfortunately, when it comes to this dating method, it is slightly destructive because we have to take micrograms of samples at least out of the rock, shiny rocks like this one. They're either slightly associated in a way that they're either above slightly or slightly below the art. So we try our best, but so far, this is what we've got. And I didn't develop it. It's actually been around a long time. I've just proven that they really do come from bacteria.
Great presentation, Faris. I'm really interested on your picture there. You've got two types of different artwork, it seems. Maybe just give us an understanding about, are they done the same way, the two different types of artwork on the rock there?
Great question. Okay, so everyone, rock art 101. So this giant kangaroo here, right, we call them irregular infill animal patterns, all right? And so basically, this is the oldest kind of art. Okay, and then if you look down here, it's actually also a bunch of flying foxes, which is really cool. So this style of art is around 17,000 years old. Okay, and if you look at the art style over here, so these are actually humans in ceremonial dress, and this style here is called the Guion style. And this art is around 12,000 years old. And we don't only use my radiocarbon dating, well, not mine, just radiocarbon dating, to date these things. We also use styles, techniques such as superimposition, where we determine which ones came first based on how the lines overlap.
All right, our final presenter tonight is Sophie Crouch with her presentation, Mystery of the Black Goo.
This is the story of a sticky black goo, so elusive that its chemical structure has puzzled scientists for almost 20 years. But to understand it, let's start with something you have heard of, dopamine. Now, dopamine's that chemical in our brain that gives us a little buzz when we finally beat that level on Candy Crush, and then suddenly it's 3 a.m. But dopamine doesn't just control how we feel. Under the right conditions, dopamine transforms. Now, these paper clips represent dopamine molecules, and as soon as I add them to some water, they start rapidly reacting to form all these new types of molecules, which all combine together to form this black clump of molecules called polydopamine. So, what can polydopamine do? Well, it sticks to absolutely everything. It's antibacterial, it's conductive, it's stable, but most importantly, it's safe for us. And that's why we're so excited to use it to coat biomedical devices, like heart stents. But for the last 20 years, scientists have asked the question, what does it look like at the molecular level? Because if we can understand its structure, we can understand why it works so well. Now, usually this means dissolving it in a solvent, like water or alcohol. Then we analyze it. It's kind of like giving it a molecular MRI. But polydopamine doesn't dissolve. It's like this clump of paper clips, just so tightly tangled that no ordinary solvent can pull it apart. So for the last 20 years, scientists have just guessed. Well, thousands of peer-reviewed guesses, but the data is hazy because it won't dissolve. So the structure remained a mystery. But that's where we came in with a bit of a cheat code. Ionic liquids. Now, these are special salts that are liquid at room temperature, thick like honey, but you definitely don't want it on your toast. Now, these are full of charged ions, which are like tiny little magnets designed to pull molecules apart. But there are millions of ionic liquids, so which one works?
Well, spoiler alert, we found it. We found the one ionic liquid that can finally dissolve polydopamine. We pulled the molecules apart, analysed it, and for the first time, we finally saw what it is made of. After 20 years, we solved the mystery of polydopamine, born from dopamine, the very molecule that shapes our thoughts and feelings, and now as polydopamine, which may shield us from harm. Isn't it cool that the molecule that makes us feel alive may very well just help keep us alive?
Great presentation, Sophie. Judges, round to you for your last round of questions.
Fabulous. The black goo has been solved. So given maybe you got $1 million, to fund your research tomorrow, what would your next step be? What would it look like?
So previously, when they used it to coat, because it hasn't been dissolved, all of the coatings have been really irregular and bumpy. And for things like antibacterial coatings and even coatings of electrodes, so I mentioned it's conductive, we need it to be really smooth. But now, because we can dissolve it, we can actually deposit the coatings like 100 times smoother. So now it's really trying to just use it for the antibacterial research and trying to actually test that and see if ours is performing better, which, fingers crossed.
Thank you, Sophie. I wonder if you can share with us what's the impact of this? What could happen knowing this now?
Yeah, so I mentioned the antibacterial properties of it, so that's one of the big areas. So one of the biggest problems is infections that happen in hospitals, so just getting things like implants, so I mentioned heart stents. So using that as a coating to prevent bacterial infections rather than the current stuff is usually silver, which can be toxic to the body, whereas this is made from dopamine. Even though it's slightly different, we know it's not toxic, it's compatible because it's already in us. So hopefully, using that instead, we can hopefully save a lot more lives.
Congratulations to all of our wonderful finalists. I have seen some amazing science on stage tonight and so well communicated. I'm going to now leave these four with a really difficult job. Our judges are going to go off and deliberate to decide on our 2025 FameLab winners. We're going to give our judges 20 minutes to deliberate. During the break, I invite all of you to go fill your glasses. Have a break. I will hassle you and tell you when you need to come back in those 20 minutes. Thank you. Welcome back, everyone, including our judges. Tricky stuff. Took your time. I'd now like to welcome the Honourable Stephen Dawson, MLC, to the stage.
Thanks, Kelsey, and good evening, everybody. I'm going to start by saying Nala Kadich Wajak Nyunga Murt Kayan Kedak Nijabuja. Can I acknowledge we're on the land of the Wajak people of the Noongar Nation tonight and pay my respects to elders past, present and emerging, and can I acknowledge all First Nations people here, and for those who are watching on screen, can I acknowledge the First Nations people on the lands where you're gathered today. Now, of course, everybody here is very important, but I do want to acknowledge a couple of people, including Coralie Bishop, the CEO of the Foundation for the WA Museum. We've got Jason Fair, who's the acting CEO of the WA Museum, Boola Bardip, to Julie Fallon, the Executive Vice President of Woodside Energy, to Professor Lindy Fitzgerald, who's here, the DVC researcher at Curtin University, and to Professor Helene de Burke-Woodman, the Pro Vice Chancellor from the University of Western Australia. Lovely to be back to the FameLab finals again. I missed last year, but I was here the year before, and how good are those communicators? Let's give them all a round of applause very quickly. Each and every one were sensational, and really I do congratulate them because they presented world-changing research tonight. And to each of the finalists, can I say your work, but also your ability to communicate it with such passion and clarity makes me really feel confident that the future of science in Australia is in very good hands. Can I also acknowledge the judges? I'm glad it was you, not me. I'm looking forward to hearing who does come out at the end, but each of them deserve to be on stage tonight, and thank you for the role that you've played. Science really does underpin every part of our lives. It drives the creation of exciting new industries such as space, life sciences and clean energy, and it also provides the solutions to environmental, health and social challenges that we face now and indeed we will face into the future. STEM communicators like our FameLab finalists are vital in building a positive culture of science in our community. By sharing your knowledge and enthusiasm, you inspire curiosity, you raise awareness, and you encourage people of all ages and backgrounds to consider careers in STEM. Initiatives like FameLab are so important because they support diverse, dynamic role models who connect with the community and inspire the next generation of scientists. And this aligns perfectly with our state government's STEM skills strategy, which is about building a strong culture of science across our state, celebrating STEM in the same way we celebrate sport or indeed the arts. And what better setting to do it than here tonight at the WA Museum Boola Bardip, beneath Otter the whale. Now, since FameLab was introduced in Australia in 2019, Western Australia has proudly hosted the national final. And I want to thank the WA Museum, Professor Lynn Beasley, AC, and all of the FameLab's past and present supporters for helping make this possible. I think Western Australia is at the forefront of science communication, and I look forward to continuing our STEM, continuing to grow our STEM skills and our culture in this amazing state. So again, to all of the finalists of FameLab 2025, thank you for sharing your important research with us tonight. You have shown us not only the brilliance of your science, but also the power of your communication, and I cannot wait to see where your careers take you. I wish you all the very best of luck, and it's now my great pleasure to welcome to the stage Coralie Bishop, the CEO of the Foundation for the WA Museum. Thank you.
Thank you, and good evening, everybody. Thank you, Minister, for your wonderful words. And thank you, Irene, for your beautiful welcome to country. I, too, wish to pay my respects to Whadjuk elders, past and present, and celebrate their culture and continuing connection to our beautiful land, seas and skies, both here on Whadjuk Buga, Noongar Budja, and for those watching online, wherever you are. The Foundation for the WA Museum is the producer of FameLab Australia, and we are proud to be hosting the final here at Boola Bardip. The Foundation's role is to cultivate and develop relationships with individuals, organisations and community groups who understand the critical role that museums play in society. We work to enhance the impact of stakeholder support and the long-term financial stability and sustainability of our WA Museum to ensure the greatest impact not just now, but for future generations. We do this because we believe museums are very special and important places for connection, discovery, and curiosity. The WA Museum advances global knowledge across a wide range of cultural and scientific disciplines. The museum is a hub for sharing knowledge with people, whether they visit one of the museum sites or explore the work online. Like the museum, the researchers you've seen tonight are all trying to solve the big questions we face for our future. FameLab is a great example of the impact the Foundation and the museum can have. Not only does FameLab provide early career researchers from across Australia with quality science communication and media training, but it enables the scientists doing research around the country to tell their stories and explain their research to people like you. A number of past FameLab alumni have gone on to high-profile careers in science communication, and it is wonderful to see the impact that FameLab has had on their career trajectories. I have to say we couldn't run FameLab without the incredible support of our partners, and again, I want to offer our heartfelt thanks to the West Australian Museum, Cheltenham Festivals, Comet Science Communication, the Community Broadcasting Foundation, Curtin University, the Forest Research Foundation, the Government of Western Australia through Inspiring WA, thank you, Murdoch University, the Wellspring Foundation, and the University of Western Australia. Can we please thank them? Also, huge thanks to our 4 brilliant judges. You definitely had the most difficult job, and thank you for your time and for volunteering. Deborah Levitt, Dr. Jennifer McElwain, Luke Smith, and Dr. Lisa Kirkendale. Thank you so much. Tonight, we've all witnessed the culmination of the 2025 FameLab program. 12 highly talented science communicators who aren't just at the cutting edge of scientific research in their specialised fields, but have excited, inspired, and sparked our curiosity through their presentations. And I have learned a lot tonight. So while this is the grand finale for our finalists in FameLab, we hope that it's just the beginning of great things to follow in their careers. And now, without further ado, the moment we've all been waiting for, the announcement of our winners. Let's welcome all our finalists back to the stage. Congratulations all. All right, here we go. Our first runner-up is Kenya Fernandes. We all fell in love with Beatrice. The runner-up is Caitlin Dudley. More mung beans. All right, drum roll. Ladies and gentlemen, the 2025 FameLab winner is Tammy Lee.
Congratulations, big congratulations to our winners, but congratulations to all of you finalists tonight. You have shown so much bravery, so much creativity in communicating your science, and it's been absolutely amazing to see how you are tackling real-world problems with some seriously incredible work. The final congratulations and thanks must go to the team at the Foundation for the WA Museum who've made this happen, including FameLab project manager and arguably FameLab icon, Wayne Lubb. And to everyone else who's been working in the background to put this together tonight. So let's have a round of applause for the whole team. And to you, our audience, go forth, enjoy your night, hopefully with new awareness of the science communication in your day-to-day night. Thank you and good night.